guys, we're going to have a shot before we get pictures of this lovely group for the prep news. Has already said what's going on. They're going to write something. If there's time.
I'm just going to get it handy.
All right. Hello, everyone. Welcome. My name is Mary Russo, and I'm a proud member of our science department at SLUH and a curriculum coordinator here. And on behalf of our science department chair, Tim O'Keefe, and our principal, Ian Gibbons, I'd like to welcome you to STEM Night. STEM is an acronym for science, technology, engineering, and math. Tonight, we'll be hearing from several of our own SLUH students on some pretty cool, robust science research or impactful programs or shadows that they got involved with over the summer or this fall. Please find the topics and the programs on the sheets on your table. So as somebody who's been teaching high school science for going on 23 years, there is nothing more satisfying than seeing students who are able to put their foundational science, technology, engineering, and math knowledge to work. Most of our presenters are current seniors, but we do have a junior and we do have a sophomore. But most are seniors and have completed at least our science requirements, biology, chemistry, and physics, and hopefully got to see the applications of these core concepts in the programs and the shadow experiences, not to mention the potential privilege of adding new knowledge or discovery to the common collective. Tonight's format is as such. We've asked veteran SLUH STEM teacher Bill Anderson to give us a primer on a longitudinal study he conducts with his environmental STEM course, followed by 6 student scholars or groups of scholars who will be sharing 8 to 10 minutes, right guys? 8 to 10 minutes presentations. Bathrooms, ladies and gentlemen, can be found at the back doors to the right, also in the lobby over here. It's a pretty casual evening, but we do have a couple of requests. Could you please refrain from using your phones? That can be distracting for the presenters, and maybe place them on silent if you wouldn't mind. And if you need to leave or go to the bathroom, could you, if you can, do it maybe in between the presentations so it's not as distracting. We've provided for distance just by leaving the tables up as folks kind of come in late and join us, and our folks on the live stream. But we do ask that you keep your masks on if you would stay masked. But the fellows, when they come up to the podium, they will be unmasking with the distance and the size of the room. We are very safe. At our practices last week, the boys went through their presentations and we gave them notes and feedback, and tonight we gave them pizza. Most of the feedback centered on this. Some of you just spent 6 to 10 weeks immersed in a research lab on a particular topic and are probably all geeked up about the cool science that was going on. However, your audience will not know about much of what you are talking about. Give them a reason to listen. And to the audience, we want to thank you for supporting our boys. If you're looking for more on STEM activities at SLUH, please go to our website. You can check it out there. Talk to a science teacher. Join the STEM club. You can talk to Ms. Wellen, Mr. Hinders, or our iLab, Jeff Schaefer, in our iLab, 5,200 square feet of wonderful innovation space, and gain access to our Thunder laser, our CNC, our 3D printers, et cetera. STEM Club does have most direct access, I believe, to that space, but you guys can get access to it. Otherwise, that said, let's get started with our first speaker. Bill Anderson is a veteran STEM teacher at St. Louis U and currently teaches accelerated chemistry, AP environmental science, and an engineering, applications of engineering course called environmental STEM. While there are many, many things on his resume I wish I could boast about, he was instrumental in working with the University of Loyola Chicago for an open source environmental text known as Healing Earth. In the words of Loyola professor Dr. Michael Shook, in an email he shared with me years ago as I was preparing remarks on Bill's behalf, which he hates, by the way, he said, "The Healing Earth textbook asks science teachers to expand their research beyond teaching their students what they need to know about environmental science and into their students developing moral values, inner spiritual beliefs about the natural world, and their motivation to act." As you can imagine, not every environmental science teacher wants to take the risk of this approach. Bill has taken on the risk, and we are all learning from him as to the positives and negatives of the textbook. In other words, Bill has been invaluable as a resource for piloting and testing and writing this textbook. There are young people in places like Indonesia and Zimbabwe right now studying environmental science because of Healing Earth and Bill's contribution. Again, that was one of his colleagues at the University of Chicago. So thanks to Mr. Anderson for kicking off our STEM night with a presentation on a longitudinal study. What does that mean, fellas out there, younger fellas? Check-ins over time. He's done this same experiment many, many different times, and he's going to tell you a little about it using data collection with our weather balloon. Mr. Anderson.
All right, thank you, Mrs. Russo. Let's see. All right, so the environmental STEM course started about 6 years ago after the science department was encouraged to develop a STEM course. We'd kicked around some ideas. One thing that I had wanted to do for quite some time was to launch a weather balloon. I thought it would be just a tremendously exciting science project for our young men to undertake. So we started kicking around some ideas. I wanted a study that was going to be a longitudinal study, which is very different from our typical science lab. Typically, a lab is kind of a one and done event. You do the lab, day or two, and then it's done and you move on. I wanted something that was going to go years, so our young men could be part of something that came before them and something that was going to come after them. I also wanted something that was of significance and relevance to our society, and I can't think of anything more important than study of our atmosphere and climate change. So it turns out one of our physics teachers knew a guy who knew a guy, and all of a sudden we're launching weather balloons. The goals of the project are pretty straightforward. I want to understand the structure and function of the atmosphere. So first and foremost, what is our atmosphere like? We want to investigate seasonal differences in the atmosphere. We want to look for long-term patterns or trends. And we also want to develop some collaboration, especially with the IT department, the mathematics department, and anybody else in the building that would be interested. This project could not happen without the assistance of John Dickman from our IT department. He's been invaluable and continues to be every year. Project always starts the same way. We have a couple of days of brainstorming and whiteboarding, just throwing out all the ideas that we possibly can about what we'd like to look for, what we'd want to measure, how we're going to measure it, what equipment we're going to need, how are we going to record the data, how are we going to get the data back after we launch this silly thing into the air. So we spend days just scratching ideas until we finally come to a good plan. We spend a couple of days programming Raspberry Pi, the cameras. We put 3 cameras on our weather balloon. We put one on the top pointing straight up, one on the bottom pointing straight down, and one that goes out laterally to give us a wide variety of pictures. Once we've got the cameras programmed, we start designing the payload, in this case, just a styrofoam cooler, and packing all of our instruments, our power supplies, our data collection devices, and getting them ready for launch. Launch day finally comes along, and everybody's got a task. You see these young men inflating the balloon that we use. Others have a variety of tasks getting the payload ready and checking to make sure that all the instruments are working. Here we see a final check of our payload, which ends up looking something like this. Basically what we have is a styrofoam cooler mounted on a wooden frame, and all of our devices either set inside the cooler or attached to various points on the frame. Once we've checked all the lines, all the ropes, all the knots, all the equipment's working, it's time to make one final check-in with the FAA and let her fly. So here you see the very last moment before the balloon takes off. Once the balloon is launched, Mr. Dickman and I hop into the van and chase it. We have 3 trackers on the device, simply because 3 is way better than 1. 1 is dangerous. One of them is just a little commercial device you can buy. People will buy them to put them on, like their boats or trailers or RVs in case they disappear and then they know where they are. Ours works great for the weather balloon. The other 2 are, it's called APRS systems. They're basically ham radio systems that'll transmit a signal back to us via ham radio signals. The map that you see here was a result of one of those ham radio signals that we collected during the flight time. So the balloon's flying, we're chasing, eventually it lands, and then we pick it up, bring it back, and we have a bunch of data to deal with. This is the core data that we typically gather: altitude, latitude, and longitude, speed, temperature, pressure, relative humidity, the percent oxygen in the atmosphere, the intensity of the ultraviolet light, and date and time. The date and time are crucial when it comes to our photographs. When we take the photographs, we timestamp each photograph. So if we know exactly what time the photograph was taken, we can figure out where it was. We'll know its latitude and its longitude, and we'll know the altitude. So that gives us a lot of information to go along with just a bunch of pretty pictures. The data is typically shared out school-wide, especially with other members of the science department and the math department. The probability and statistics course and our new course in mathematics and data science are all using the data from our launches. The students spend quite a number of days analyzing the data. Typically, we'll use Logger Pro or more commonly Excel to produce graphical representations of our data. And many of the graphs turn out exactly like what they expect. So for instance, with pressure and altitude, they know, well, they think they know going in, and they're right, that as you go higher in the altitude, the pressure gets less. And that makes sense, and this graph is exactly the same as our oxygen graph, because as you go higher, there's less air, fewer molecules, so the pressure goes down. Some of the data, they don't know what to expect, and wind speed is a good example of that. If you look, the wind speed starts off relatively low at ground level, rises until it hits a maximum, comes back down, and then frequently, but not every time, will rise again at a higher altitude still. What's interesting is to pay attention to that particular spot where we see the maximum winds, and that'll come up again in a moment or two. One of the pieces of data always throws them for a loop, and that's one of my favorite ones because they don't expect that to happen. If I were to ask all of you in the room what happens to temperature as you go up in altitude, I know every one of you would say that the temperature decreases. We see snow on mountains, so we know it's colder the higher you get. But when you plot the data, this is actually what you find. The temperature does decrease up to a certain altitude. Then there's a period where there's a little bit of a pause in that pattern, and then we see the temperature start to increase. And what this shows us, and what this tells us, is that our atmosphere is divided into layers. That lower portion where the temperature is decreasing is our troposphere, where all human activity essentially takes place. And then there's that pause, that's called the tropopause. And then where the temperature increases, you've moved into the stratosphere. And I think that's one of the things, and I'll say this a number of times, that's just absolutely phenomenal about this project, is we've got a group of ragtag high school juniors and seniors and myself, I include myself in the ragtag, and we're able to take a device, it's not a big fancy glorious device, and we're able to launch it out of the lower atmosphere, and in this case, over 100,000 feet up into the stratosphere. It's just absolutely phenomenal. And this is the 11th launch we've had, and each one of them is every bit as exciting and exhilarating as the previous ones, because this is just absolutely phenomenal. What's really interesting is when we start comparing some of the data we get. Here we see on the left the temperature in blue and the wind speed, the more scattered points to the right-hand side. And if you look carefully, you can see that maximum wind speed occurs at just about the same altitude as that change in the temperature pattern. And we've seen that every time. And that high-speed river of air that we've got at that maximum speed is the jet stream. And that jet stream always occurs just below the top of the troposphere, so just before the tropopause and the stratosphere. And we see that every time. Gets even more interesting when you look at it year after year after year. So we've got a graph like the one on the left of temperature change. And what we've done is we've plotted each of our 11 launches and the altitude at where that temperature change occurs. And it goes chronologically, summer, winter, summer, winter, summer, winter, all the way across. We launch twice a year, so we can get two discrepant seasons. And you can see that the summer launches are always significantly higher than the winter launches. So it's awesome that we've gotten to a point where we've got enough data that we can start to see some of these long-term patterns that a longitudinal study would allow us, that a one-time launch never would tell us. What this tells us is the height of the troposphere varies. The troposphere is lower in the winter than it is in the summer. That in turn affects things like wind speed. Wind speed is much higher in the jet stream in the winter because the troposphere is lower and the rivers of air have a much greater velocity as they flow towards the polar regions. One of the best things is the photographs. They're just always awe-inspiring. This one I particularly like because it shows our balloon on the left just at launch, so just moments after we launched, and then at its maximum altitude. You can see the significant change in volume because the pressure has decreased as it rose. Just after that picture on the right at maximum altitude, we capture this picture. We get to a point where the pressure inside the balloon is so great and it's expanded to such an extent that it can't expand anymore and it ruptures. And we're always shooting for a shot like this to show the balloon and the shrapnel. And the really interesting thing is the background. The background is black. The background is black because that's space. So that's the background to our photograph is space. Once the balloon pops, our parachute will deploy and it brings our payload back down to Earth relatively smoothly and hopefully not in a tree or a river or things like that. Although trees are their favorite landing spots. We get other good pictures like this. So here we see one where I've got an altitude and a location, and we do that because we timestamp them. Otherwise, we wouldn't know where these pictures were taken. These are always my favorite pictures. This was taken just a little under 90,000 feet, and it's just absolutely spectacular. Again, this is just a group of high school young men, and we've captured a picture that shows that transition from atmosphere to space, that clearly shows the curvature of the Earth, and we're able to capture that, bring it back, and share it with their classmates and whoever else in the community is interested. So pictures like this are always just so awe-inspiring. If you look at where all of our launches, and I realize this graphic is pretty small, we plot all of our landing locations, and you notice that all of our landing locations for the winter are much further to the east. So Again, because the jet stream is stronger in the east, it's going to blow further. To wrap things up so our boys can get their turn, the things that we're really looking for, the data collection, the knowledge, the science learned is awesome. The pictures are phenomenal and really awe-inspiring. Can't say enough about the magnitude of this accomplishment. We've got these young men have an extremely rare opportunity to venture that far into the atmosphere. It's just mind-boggling. And I'm amazed myself every time that we can pull this off and that we get the data and we're seeing the relationships and the patterns that we're supposed to see. It's just an incredible project. Teaches them that science is a process. Okay, we learn something new every time. We've got a core set of data that we gather. We've tried other things. Some things have worked, some things haven't. So you learn and you adapt, you modify. We learn from our failures. The very first time we launched was horrible. We checked all our equipment, filled the balloon, had it ready to go, called the FAA, let it go, and it sat there. Didn't move off the ground at all. So obviously we'd miscalculated. Redo the calculations, rethink it, take it all apart, fill it back up again, and then it took off just like it was supposed to. But that's science. You do something, you hope it works. If it doesn't, you fix it and you try it again and you keep trying until you get it right. And then lastly, when you've got an opportunity, please don't ever hesitate to say yes to it. I think our young men would agree with that, with the opportunities that they've had, but it's definitely true for me with this weather balloon project. It has just been literally the most exciting thing that I've done here in my whole career has been here, but it's been the most exciting thing that I've ever been involved in the science department here at SLUH. Thank you very much. I'll be around for a while afterwards. I've got some of the stuff. If you want to take a closer look at it or ask any questions, feel free to do so. Thank you very much.
Thank you, Mr. Anderson. Like he mentioned, guys, if you want to hang out afterwards, we're going to go ahead and kind of push forward with, but you probably should have all sorts of questions. I have more questions. I sit right next to him in the science office. I have all sorts of questions. Every time I have all sorts of questions. All right, our first student presenter is Angad Gathra. Angad, go ahead and go up and bring up your slides. Angad is a senior, and he's going to be talking to us about research that he did through a program called STAR, Students and Teachers as Research Scientists.
My name is Angad Gathra. I'm a senior at St. Louis University High School, part of the class of 2022. And I was one of the 3 students at SLUH High who participated in the STARS program. So what STARS program is, it allows 36 students, it exposes 36 students to STEM-related research. The schedule is something along like, on Tuesdays and Thursdays, we listen to various doctors and professionals in the STEM-related field, and we listen to their experiences and what they usually do in a typical day. And then on Monday, Wednesday, and Friday, we did research, and then we ultimately submitted a research paper. And so I was actually lucky enough to be assigned to a wet lab, so I was actually able to go to a professional lab and view what my mentor did and what I was researching for the summer. And I was paired with Dr. Herbie at the Herbie Lab at Washington University in St. Louis. So neurofibro mitosis type 1 is something that is really related to malignant peripheral nerve sheath tumors, which is my topic. NF1, or neurofibromatosis type 1, is a genetic disorder and is estimated that 1 in 3,000 people are affected by NF1. Common signs for NF1 include dark spots, freckles, and neurofibromas. And what makes NF1 so dangerous is that 50% of diagnosed cases occur without family history. And so there's a 10 to 13% chance that a patient with NF1 can develop a malignant peripheral nerve sheath tumor, or MPNST. Treatments for MPNSTs include surgery, radiation, and chemotherapy. However, they've been found to be rarely effective, as MPNSTs reoccur in about 50% of patients. So there are many clinical barriers to MPNSTs and treating MPNSTs. First one is most patients with MPNSTs die within 5 years of their diagnosis. So it's really hard to accumulate data over the patients with MPNSTs. And also, it's really hard to detect if the MPNST is malignant or not, because MRIs cannot detect malignancy. And so that's what makes instituting an effective therapy so hard. And so there's a pressing need to develop better biomarkers of MPNST development. So as you can see in this graphic, between part B and C, that's when the Schwann cell first becomes malignant, and there's no biomarker between the B and C modules. So what we're trying to currently do is find better biomarkers to detect malignancy. So TYK2 is a regulator in MPNSTs. Through previous genomic profiling at Herbie Lab, we found TYK2 to be frequently mutated and overexpressed in a subset of MPNSTs. So you might be wondering, what is TYK2? TYK2 stands for tyrosine kinase 2. It is a non-receptor protein in the JAK-STAT family, and has been shown to regulate apoptosis, cell proliferation, and invasion. And TYK2 basically mediates cytokine signaling. So as you can see in this graphic, the cytokine interferons, so there are 3 types of interferons, and once they bind to their respective receptors, it will actually activate TYK2. So TYK2 is under the cell surface, so it'll activate TYK2, and then through STAT proteins, the STAT proteins will then enter the cell and then bind to the promoter region of the DNA, and that will release some 300 interferon stimulating genes. So this is basically the purpose of my research over the summer. The first 2 questions is what the lab is currently trying to do, and as a whole, what they're trying to do over the summer. So they were looking at, can TYK2 serve as a biomarker for MPNSTs? And then, are TYK2 inhibitors a viable strategy in MPNST treatment? And then what's in the red box is basically what I focused on. So I tried to dissect out pathways through which TYK2 functions. So I looked at downstream things of TYK2. And so what I looked at 88 genes of interest through qPCR, and then I kept 8 housekeeping genes for a control. So this is a little bit about my experiment. First, I cultured MPNST cells. They were actually donated to WashU from Johns Hopkins University. And so I cultured that in a fume hood. So at a lab, you'll basically be assigned to a fume hood, and you have to be really disciplined over what you can take into a fume hood because you want to preserve sanity. And so once I cultured these MPNST cells, I actually treated them in a 6-well plate. The first 3 wells, as you can see in this graphic, were actually kept as a control with DMSO. And then the bottom 3 wells, I actually treated with WU12A, which is a TYK2 inhibitor treatment. After that, using a RNA mini spin kit, I was able to isolate the RNA. And then from there, I was able to synthesize cDNA, and that is where qPCR comes in. So a qPCR plate typically has 96 wells. You fill each of the 96 wells with cDNA plus cybergreen premix, which all comes in the set that you order. After you fill each well up, you put that into a thermocycler. So the thermocycler will first denature the cDNA, and then it'll anneal it, and then it'll elongate it. So basically the cell is amplifying, and as you can see in this graphic, to your left or to your right, the cells are basically being amplified, so you can see the amplification charts. And so taking AP Stats actually really helps. That's a class that you can take as a junior at St. Louis University High School, because we were able to, I was supposed to know what confidence interval and p-values were, and I was able to learn that in Mr. Hannick's class, and that all came in pretty well. Just knowing that helped me so much over the summer. And so after getting these amplification curves, I was able to detect which of the 88 genes were upregulated and which of the 88 genes of interest were downregulated. So in the graphic to your left, you can actually see the bottom part of the graphic. You can see the 88 genes that I looked at, and then the top part shows the correspondence of if it's upregulated or downregulated, and this is all through gene expression. So there were 88 genes, and numerous genes were found to be upregulated upon TYK2 inhibition. And some of the upregulated genes included IL-6 and the IL-6R genes, and that's involved in the IL-6 and the IL-6R pathway, which are related to cancerous pathways, and those were upregulated. And that kind of shows how MPNSTs and TYK2 are so complex, because when you inhibit that, you get this upregulations of other cancerous pathways. But also, numerous genes were found to be downregulated upon TYK2 inhibition. So some of the genes that were found to be downregulated were the P13K, AKT, and the mTOR, and those are all associated in the P13K, AKT, mTOR pathway, and that's related to cell proliferation and cell survival. Some of these genes actually are really fun to name, like SOS1 and SOS2. It stands for son of seven, and they're just really cool. They're really random too sometimes, and it's just really funny just getting to know some of these gene names. And so like I said, my role was to dissect out TYK2 pathways in MPNSTs. So I was actually able to use the KEGG pathway, which offers a predicted pathway based off of gene expression that you can then later test with specific inhibitors and gene knockdowns after you validate which genes are actually involved. So like you can see in this graphic, when there's an upregulation of cytokine and hormones, you can see that there's a downregulation in the STAT protein, the SOS protein. And so each of those genes, like I said, are related with different types of roles. They control apoptosis, cell proliferation, and cell survival. And then later, after that, towards the end of my program, the end of my 6 weeks, we validated the genes of interest using qPCR, because you always want to validate biological significance, and that's very important. And so this leads me to my future directions. So we're going to continue to validate gene expression for other genes of interest, like PIM1, B2M, CNTF, and other genes. And so what I basically did over the summer is I looked at gene expression. The lab currently is looking at protein level expression. And then later, we can use that data to dissect out possible pathways by which TYK2 functions in MPNSTs. And so at SLUH, right now I'm taking AP Bio and AP Chem in the science field. And the STARS field kind of shows the synthesis between the two. They're really applicative. A lot of things that you learn in AP Bio are related to the things that you learn in AP Chem. And so the synthesis of like how the two come together is just amazing. And you'll experience that a lot throughout your years between many more subjects. And one of the most important things I want to highlight, especially working on a team in a research lab, you have to have collaboration. You have to work as a team. If you don't understand anything, you have to have a voice and tell them that you don't understand it so they can help you. So at WashU, I didn't understand some of the concepts, and I just asked my mentor and the couple of undergraduates that I was working with, and they gladly helped me. And so that's very important, just working together. And SLUH does a great job of creating this bond and brotherhood between your classmates. And lastly, I'd just like to give a special thanks to Dr. Herbie, Dr. Borcherding, Neha, and Dr. Warndl, my writing mentor, because they all helped me tremendously. Thank you.
Thank you, Angad. I want to remind some of our younger guests and folks who are watching on the live stream, Angad is a senior. He's a senior. So did you understand all of that? No, and that's okay. Listen to it, because fellas, it'll be you. 5 years, 6 years, you'll be surprised. You'll be surprised. Keep the faith, keep working. Let's go ahead and hear from our next presenter, Xavier Jallow. Xavier is also a senior, and we're going to change up. He did a little bit of a shadowing experience with a mobile health clinic. Was it not this one, right? Was it this one? It was this one, wasn't it? I put everything on my machine, and they're not used to my machine. So let's see. You're on.
Good evening. My name is Xavier Jallow. I'm a senior at SLUH. I'm involved in ACES and its affinity groups. I'm also involved in STARS. I'm a leader of the One World Club, and I'm part of the Medical Careers Club. So what I did over the summer on June 28th, 2021 is I shadowed at a clinic out in Cadet, Missouri called the Rural Parish Clinic. I'm just going to give you a little bit of backstory on how I was able to get the shadowing. My mentor, who is Ed Rata, who is a retired SLUH lawyer, actually set me up with a pre-med advisor from WashU, who is a PhD MD candidate at WashU. And somehow the conversation at dinner landed on what steps I'm taking towards the pre-med track and what I would do in college and high school and beyond. And one of the components that I was missing with my explanation was the part of volunteering and service. On your resume, when you are applying for medical school, you have to show that you are a compassionate and basically a human being that has people skills, not just book smarts. So knowing that now, I actually sat down over lunch with my mentor's primary care doctor, and he basically was thoroughly impressed with me, and he eventually offered me the opportunity to shadow at the Rural Parish Clinic. So the Rural Parish Clinic is a program through the Archdiocese of St. Louis that serves uninsured people in two types of ways, through medical and also dental. So the van that I went out to was in Cadet, Missouri, and I actually went on the medical van. I didn't get the chance to see the dental van, but on the medical van, we have none of the modern medical imaging techniques that we have at hospitals, like your angiograms, your PET scans, your CT scans, such. But we did have one small ultrasound machine that we really didn't use too much. But yeah, it's nothing like the whole philosophy behind the program is you're as poor as the people that you are serving. So if you went 30 cents over budget, you would have a big problem with Sister Marie Paul Lockhart, who heads the program. So on the medical van, I actually, we got to see a plethora of cases, but mainly what the medical van does is they really serve acute care like your cold, flu, and minor injuries like broken bones and infections. But they also do physicals and also psychiatric like depression and anxiety, and also chronic diseases like diabetes and heart and pulmonary diseases. So an example of a patient that you would see on the van would be this first lady who came in because she was having panic attacks, and also she had trouble breathing. So in her past, she had something called hyperlipdemia, which is basically a collection of fat or lipids in the blood. And she also had a hypertension, and in both of her family lineages, they both had a panic disorder. And she also had 89 points over the ideal cholesterol. So the diagnosis or the treatment for that was an increase in the medicine that she was receiving, which is simvastatin, which is a cholesterol-reducing drug that lowers your chances for a heart attack or a stroke. And she was also told to cut down on the coffee from 3 to 1 and cut down on the acidic foods and avoid late meals. Another patient that you would see, this guy, he came in, he was a cook. He, prior to coming in, he had neck pain, and this was a repeat visit. But I think at his job, he actually slipped on a pan before, and that is really important into the diagnosis, because if you were to go to a hospital, they would probably order a functional MRI or MRI to swiftly diagnose this. But since we don't have that type of equipment at the clinic, we had to use some type of other methods to diagnose this. So he came in because he had a headache, neck pain, and paresthesia in the fingertips. Paresthesia is basically a pins and needles numbness feeling in a region of your hand or your whole hand. He actually had it, if you can look at the graphic right here, he actually had it at C8, which controls this part of the hand, this region of the hand. And there's also other graphics that shows like the intervertebral disc and the nerves control at C5, C6, C7, and C8. But what his diagnosis was a pinched nerve at C8, and he had to do some neck exercises and also buy or get an orthopedic pillow or just basically roll a towel and sleep on it in his pillowcase. So I'm going to further explain how you would get a pinched nerve. So what happens is there's an external pressure that is exhibited on the intervertebral disc, and that causes it to herniate and bulge out and come in contact with a nearby, with the spinal cord or a nearby nerve, and it produces an effect, whether that be paresthesis or another like paralysis in the arm or all over the body. It's really infinite possibilities. So this was a mini med lesson that I got from Dr. Tom Johans, who was an anesthesiologist that also served on the clinic. We talked about everything from the blood flow in the heart to ischemia, which is a not enough blood going to a particular region in the body. We also talked about Laplace's law, which is a way for doctors to predict the pressure in blood vessels. We talked about adrenaline and acetylcholine and how they come in and interact with neuroreceptors. We also talked about glucose and lactic acid and how they interact with each other, and basic potassium pump, etc. It was one of the most fast-paced lessons I've ever received, and it was over lunch in like 30 minutes. So it was pretty crazy. These were all the questions that I had after the shadowing, and they range from freshman bio, what's the difference between type 1 and type 2 diabetes, to what are varices, what is hematemesis, what is macrocytosis, what is a mitral valve prolapse. All these things that you will cover in SLUH, hopefully. And I found myself kind of rethinking and just knowing that there's no stupid questions when you're shadowing. The doctor will answer them, and he will answer them thoroughly and concisely. So my biggest takeaways from shadowing, my first one would be probably insulin prices. Since I was talking about earlier that a prerequisite to get on the van is you have no insurance. So you can't be seen if you have any insurance. You have to have no insurance. So normally, if me or you would probably go out to the hospital and we have diabetes or something like that, the insurance will cover a portion of the costs for insulin, and you would pay some out of pocket normally. But since we're talking about the uninsured, they have to pay fully out of pocket. So a question that I brought up to the doctors is, how are these people receiving insulin out of pocket if they can't even afford the basics of insurance? And I kind of pondered and asked them, and an answer that they really came up with is, Insulin is as high as it is because the pharmaceutical companies make it that high. It takes billions and billions and billions of dollars to pass the patent for drugs like insulin. So while the patent is in effect, they make their money back and more in tenfold. So it kind of, after that, it kind of gets into, is it morally good because we have the drug on the market, or is it morally wrong because it's more expensive? But I'm not going to get into that. My another biggest takeaway was, can we provide free and thorough health care without compromising the quality of service that a patient will receive? And my answer to this question is yes and no. Yes, because we can do things like the doctor did with diagnosing the pinched nerve at C8 with a bobby pin, but that is kind of a runaround way of doing it when we could have just did an MRI and got it done swiftly. But in modern times, since hospitals are kind of overloaded and have to see a lot of patients, we have to resort to these medical imaging techniques to get patients out swiftly, fast, and thoroughly. So I think it is a necessary evil that we have to deal with right now, but in some cases, we are leaning a little bit too much on your MRIs and CAT scans and such. My third biggest takeaway is that this is the career choice that I want to follow. I think this kind of reaffirmed my aspirations for medicine, and I would just like to thank the teachers at SLUH and the faculty and everyone who's kind of propelled me into this position to show off what I know. I would like to say special thanks to Dr. William Guile, the medical internal care specialist that I shadowed, my mentor, Edward Rata, and Dr. Tom Johans, who is an anesthesiologist. Thank you.
Thank you so much, Xavier. They don't have to take any science senior year at St. Louis U High, but a lot of them opt to raise your hand, seniors, if you're taking science. I know you have to. So, Xavier, honors anatomy and phys, it would make sense. Is that what you're doing this year? Very, very good, very good. How about a stand and stretch break? Can you guys stand real quick? Why don't you stand up, guys, and kind of stretch a little bit? Would you mind? I feel like we've been sitting for a bit. While you're standing and stretching, we'll go ahead and get ready for our next presenter. Mr. Fred, no, that's Robert. Robert, you're next. Robert Mize. Robert Mize is a senior. He also did the STARS program. So we'll hear a little bit about his work with students and teachers as research scientists.
Am I good to go? Okay. Like Mrs. Russo said, my name is Robert Mize. I'm a senior at SLUH. And I worked in association with the Zhang group at WashU through the STARS program. So my lab, I worked with Dr. Zhang, or more specifically, I mostly worked with his graduate student, Jingyao Li. And I spent 6 weeks working virtually on a research paper about a review of amyloids, which essentially combines the knowledge of past professors and researchers' papers into a succinct article that another researcher can read and use for future research. So I did that virtually and not in a wet lab. So I'm going to talk a bit about what amyloids are and what exactly they're composed of, what they do, and just a general background of amyloids. So amyloids, since they've been discovered in 1954, they've usually been associated with diseases. The most common one you'd see is Alzheimer's. And Rudolf Virchow first used them to describe a tissue abnormality. So they've been extensively studied since then. And in about the 1990s, the narrative around amyloids started to change. And it became something where amyloids could serve in synthetic biology instead of just being associated with disease. So I should get into what an amyloid is. So an amyloid is a protein structure, and they're noted particularly for their stability and their ability to self-assemble. So this process of simple self-assembly, it lends itself well to create tailored materials, especially structures or like a template for another material in, for example, like bio-nanotechnology. So they also serve in nature. So they're somewhere between a secondary and a tertiary structure of a protein. So a secondary structure of a protein is a local structure of the protein backbone, which is stabilized by like intramolecular and intermolecular forces of hydrogen bonding. And they are usually folded locally. And the two most common forms are either an alpha helix or a beta sheet. Amyloids are made up of multiple beta sheets. They aren't quite as complex as a tertiary structure, which is, for example, would be a globular protein. I don't have a picture of it, but this depicts multiple different amyloids, which are made up to 50,000 beta sheets. So they are quite complex structures, but they're not quite tertiary protein structures. So I'm going to give an example of first a natural amyloid being used functionally, and then I'll go into artificial amyloids that are either engineered and changed in a lab to serve a purpose in bio-nanotechnology. So in this case, a natural amyloid is used in CO2 capture. The picture shows this amyloid in different levels of magnification. So that is condensed into a powder-like substance. The C shows it from a couple of different angles. And so this is a hexapeptide, which is VQIVYK. So basically the amyloid would, with these alkylamine groups, they reversibly bind to carbon dioxide through a process called carbamate formation. And that this can essentially capture carbon dioxide and can be transported or moved. And then when heated, this amyloid structure could release carbon dioxide. So this could play a role in the environment and climate change, especially with pollution. So I'm going to get into artificial amyloids now, which, for example, I'm going to use the stabilization of graphene. So graphene is incredibly useful in bio-nanotechnology because of its different structures, and it's just a generally useful compound. But it is hydrophobic, which is a functional issue for graphene. So amyloids offer a solution to this by essentially acting like a bone would, except instead of a collagen structure, it would use an amyloid fibril. And this is inexpensive, it's degradable by enzymes, and it's highly conductive. So I should probably also speak to what a bio-nanotechnology is, because you'll hear me use it throughout. So it basically includes any devices that are engineered on an atomic, molecular, or macromolecular level. So this technology is it's used in disease diagnosis, molecular imaging, and targeted drug delivery. So another application of an artificial amyloid is in the control of gold nanomaterials. In this case, gold is quite similar to graphene in that it is used in bio-nanotechnology. It's used because of its optical, catalytic, and conductive abilities. So in nature, proteins often act as templates, which it acts as here for gold, except it's engineered in a lab instead of in nature. So this graphic kind of gives us a representation of how they form a gold nanocomposite. So it shows how our protein was mixed with deionized water. It's then incubated. It's then prepared at 2 different pHs. And through the variation of pHs, it can form different structures of amyloids. And it can essentially form different compounds and structures for the gold nanomaterials that can be used for different purposes. So I can... sorry, I cannot zoom in, but it essentially shows the distinct structural differences between these 2 forms. Well, I guess I should get into why like amyloids are a good example of synthetic biology. So amyloids essentially are, or they were never thought of in science as something that could be used for such like variable purposes. And they really were only thought of for diseases. And amyloid research really picked up in the 1960s, but it still took another 30 years before anyone really thought of using them for synthetic biology. And even today, every year they add on new amyloids and new ideas for ways that amyloids can be incorporated into this field. So a review essentially of amyloids offers new scientists the most updated set of information about amyloids that can be used for their own research. So that is the essence and purpose of a review, is just to basically have a set of data that anyone can use and can have access to, and then they can use that for their own research and build on further to allow for greater use of amyloids. So kind of why I did STARS and what I got out of it. So I got out like a greater understanding of scientific research because I had never really worked in a lab or with an actual lab before. It also offered insight into dozens of speakers, from experienced doctors to a dual MD PhD student, and that kind of gave me a lot of good ideas on where I might want to go in college and beyond. It also gave me connections with other students interested in similar careers, which would be more prevalent in person. Ours was mostly online, but I still got to meet people through smaller groups, and that was really important to me. So it also is a chance to be responsible and independent, because in this project, the only hard deadline that you have is the final paper. So like every week, they kind of check in with you, but if you want to, you can postpone your work a bit, or you can, if you're ahead of schedule, behind schedule, you can kind of set your own schedule. And so that took a couple of weeks for me to really figure out and to get a hang of. And I think this is just such a unique opportunity, because you can't have 6 full weeks of scientific research and scientific writing and just the experience of just going through it and editing and re-editing and finding new sources in a classroom, simply because there isn't enough time for that in a year. So this is just a unique project that you can only really do here, or you can wait until you're in college. So the last thing I have really is my references, and I'd like to take a chance to thank Xavier Jallow and Dr. Zhang at the Zhang Lab.
Thanks, Robert. He was referring to procrastination. Anyone else a procrastinator out there? Not Robert. He got it done. He got it done. Our next speaker, we're going to go ahead and hear from Freddie Locks. Freddie is actually a junior. So breaking the mold here, and he's going to talk a little bit in a different way about drug therapies, but fundraising for drug therapies for populations without access. Oh, you got it. Wait, hold on. Click the wrong one. Let's see. There we go. There it is. Bam. Okay. Yeah, that one. Click. No, it should be that one. See if that goes for us. Yeah, go up there. No? Yeah, you can. Here, let's see. It's not looking for us on here. Sometimes it's my clicker. It's not working there. That's weird.
So we can go slideshow present.
Yep. Go ahead. Presenter.
There we go. Perfect. Okay, so hello everyone. My name's Freddie Locks, and I'm a member of the class of 2023 and a junior here at SLUH. And today I'm going to be talking about global health and facing health inequities across the globe. I originally became interested in pursuing STEM at SLUH because I was very interested in medicine and global health. And after joining SLUH's Medical Careers Club, I got to hear from many speakers and doctors who worked within these fields. At one talk, I heard from a local pre-med advisor who was working at WashU, who had been involved in a project with a local nonprofit called Empower Through Health, which works with a health clinic in Uganda to provide life-saving medical access to citizens of Uganda who don't have that type of access. So after sending a few emails to this professor and becoming more familiar with her work, she actually invited me to work with a couple other high school students, along with a few WashU students, to help to fundraise for this clinic in Uganda. And quickly after joining this project, I learned that the health issues facing Uganda were not similar to the health issues facing us Americans or even many people in Western countries. Many of the diseases that were prevalent in Uganda were completely preventable, and many of them were non-communicable diseases. So these were diseases people got from just simply drinking water, from planting seeds, and doing other various activities just to survive. And so specifically, WashU's chapter of Empower Through Health worked on preventing a disease called schistosomiasis. And schistosomiasis is a parasitic disease which usually you become infected with through infected water. And so it's actually really prevalent in a snail population. And these snails, they're sometimes prevalent in soil, but mostly they're prevalent in the water sources in Uganda. And these snails release larvae, and those larvae actually, when Ugandans consume the water, infect the human, and the human becomes a host for those larvae. And so this leads to bladder cancers, a lot of problems for children whose bodies are too small to handle a parasite. And so just learning about this terrible disease and learning that many Ugandans just living their lives, drinking water, bathing in the river or lakes, were becoming infected with this. I knew that I had to really step up and help with this project. And so quickly I learned, almost to a saddening extent, that this disease, schistosomiasis, was actually 100% preventable because of a major breakthrough with a drug called praziquantel. And so praziquantel was a drug developed, I think it was in the 1950s, and this could completely rid the body of a parasitic infection. So once someone takes a full course of praziquantel, and you can see the drug listed here on the screen, when someone takes a full course of praziquantel, that infection completely goes away from their body. So whatever parasite they're infected with, they're completely rid of that infection. And so this is a major breakthrough for children, for adults who want to keep on living their lives, consuming water, planting seeds, and not have to die by doing those actions. And so what this WashU chapter focused around was fundraising for Ugandans to have access to this drug, because it was actually a very cheap drug to buy and distribute in Uganda, but there was really just no one taking the effort to buy and distribute it to these citizens. And so, as I worked with these other high school students and WashU students, we had to adapt to very unique methods of fundraising, because to get a high schooler to fundraise, to get a parent to fundraise, you really had to convince them that the work you're doing and the money that they're donating is impactful. And so I'm really glad of the work that we did, although it was very challenging to do all this over Zoom during a pandemic. We were actually able to create a website which in and of itself educated students and adults on the impacts of schistosomiasis in Uganda. And we did that through a blog where we interviewed children and doctors in Uganda on their experience with the disease. And then we also created within this website a unique fundraising tab where students and adults could buy different products that were created by people on our team, and 100% of those profits would go to this schistosomiasis effort. And so this is one example here. We had a graphic designer on the team who actually volunteered to draw these stickers. And so students could buy these stickers, and after buying them, they could put them on their laptop, put them on their water bottle, And not only would they be donating to help this health clinic in Uganda, but also through presenting these stickers to the public by putting them on their laptop and other places, they were educating people about what Uganda was, culturally what Uganda was, and then culturally what Ugandan citizens were like. And we also included a sticker that has, if you can see here, it says ETH. That's short for our group, which is Empower Through Health. So another way of spreading the word about this action. And so what I learned from working on this process was that because I took the risk and reached out to these people and joined this experience, I gained so much experience in global health and really just experience in STEM and leadership in general. I now plan to, in the future, take a trip to Uganda. I'm actually working on an internship right now where I'm going to go to the very health clinic that I was fundraising for to help provide Ugandans with that essential drug, praziquantel, to help save many lives. And so I think my biggest takeaway is that your ability to reach out and use the resources that are there provided in front of you is very powerful. And that's going to determine the type of experiences that you have. So as you think about your future here at SLUH, think about your future involving making the most of what you are passionate about. And also think about using what SLUH has to offer to reach your highest potential and make a positive impact on this world. Thank you.
Thank you, Freddie. So junior year, you're doing AP Physics and got it. Very good, very good. Thank you so much. We've got 2 groups left to present. I just want to remind you guys, if anyone needs to hit the bathrooms out here and out in the hallway. The 2 groups that we have left to present, we'll kind of leave a little bit of some of the biology and medicine side of things, and we'll move towards some of the physics and then some programming and some internal kinds of things that we have happening at SLUH with robotics, et cetera. So your patience is something we're so appreciative of. Why don't we go ahead, Richard Taylor, we're going to hear from next, a senior. He was our 3rd STARS student and teachers as research scientist. He did that program, and we're going to hear a little bit about his work.
Okay. I'm Richard Taylor, and like Ms. Russo said, I also did STARS along with Robert and OnGod. First, I just want to talk about a little bit like why I got involved in the program. So by the time I applied, which is I think around April, I already had around 3/4 of physics at SLUH, and I really enjoyed physics along with all of my other math and science classes that I had up until then. So I kind of knew I wanted to be a physics major in college, and I knew, and I still know that a large part of that major is going to be research in undergrad. So I really wanted to get a leg up in research experience before I entered undergrad. And then apart from just kind of getting that experience is also thinking more creatively, which I think is not something that always happens in the classroom. You're sort of just taught something and you understand it, but you're not really thinking on your own and thinking about your own ideas. And then also just getting better at scientific writing, which is similar and different from writing in English classes. So that's also getting experience with a college professor. So just some practical things as well as getting experience. And now I want to get into sort of some of the research I did. So I remember like a couple weeks before the program, the professor that I was ended up working with called me, and he started to talk about like topics that I could do. And he had a thick Russian accent. So he said, something like, would you be interested in doing time? Something about time. I'll send you my presentation on it. I was like, time, like what? It's definitely more abstract than Robert's and Ongod's presentations. And so, he sent me the PowerPoint that he had prepared for me to look at. And that night I was just looking at it, and, it didn't really make a lot of sense. It was really foreign. So I don't expect you guys to get it immediately. I think it's definitely something that has to sort of sit in your head for a while before you can start to think about the idea of time. So I think the starting point is like the idea of an event as just any sort of change that occurs. And then you can categorize events by, if they're periodic, if they're reversible, if they're if they occur over a distance, or, and then this next category is kind of how you get that transition into the idea of time. Like, does the event occur at an instant, or is it like a sequence of events? So, we think of time as just like the number on a clock, but before, that wasn't always that way. It wasn't always just a number that, oh, what time is it? It's 7:41. Before that, it was much more of a fluid concept. So you can build up the idea of time from events, time as a sequence of events, and as a numbering of periodic events. So, the sun rises and sets. So that's a periodic event, and then you can use that as a basis for using time as a number. So you can map the events one-to-one onto numbers, and then you can get what we think of as time as just a number. And then in different domains, you have different events. So then that leads to the idea that time has a different concept in different domains. So in physics, if you think about physics broadly as kind of changes in position of let's say large celestial bodies, not getting into the quantum level, time, events are changes in position that span distance and that have multiple observers. And so the time then is global because the events are changes in position. So the events are global. So the concept of time can be considered to be global. But in chemistry, the events are changes in composition. They're not they're not spanning a distance. And so the time is local and kind of intrinsic to that location. So some similarities between time in physics and time in chemistry, which is what the main topic is about, is that they both have the same origin, the same kind of building block of an event onto a numbering of those events to make kind of a quantitative understanding of time. And then, that clock time can be used across domains. You can use a clock, an atomic clock, which would be based on chemical time, to measure an event in physics. Then differences have to do with the events in physics and in chemistry. So in physics, the events are changes in position. In chemistry, they're changes in composition. So in physics, time is global, and in chemistry, time is local. And so what this kind of leads to is how we can understand symbols in equations. So a lot of physics, at least, is, you're given an equation and you have to understand what that equation means. So these equations are basically the rates of the reaction that's occurring in these reactors. So on the left, this reaction is kind of a standard chemical reaction. It's, there's no change in position. It's just changing composition. And on the right side, it's kind of a special case, or maybe a non-typical reactor where there is change in position. So the time here is not the same time, right? So this is represented by this symbol. This time is not just clock time that's measured in the lab, maybe. it would be derived from like distance over flow rate. And so, from this equation, if we didn't have that conceptual basis of what are, how can we have different ideas of time, then these equations would not be distinct from each other at all. And then my research kind of went into more like subsets and not directly related to the ideas of time, but so first the ideas of like non-steady state and steady state reactions. So non-steady state would be all these concentrations are changing with respect to time, and steady state would be that they're all remaining constant. So on the left side of this vertical line, it's in non-steady state because these concentrations are changing, and on the right side it's in steady state. And so typically everything is changing, but a big question is what doesn't change? I mean, in introductory physics classes, a lot of it revolves around conservation laws. So energy conservation, momentum conservation, and that provides constraints that you can use to solve a lot of your problems in the class. So, we have the simple mass conservation law where all the concentrations are constant, and this doesn't depend on the type of reaction. We can also look at kind of a battery of reactions where the ratio of concentrations is constant. But can we have, in these, it's kind of all non-steady state and then steady state. But can we have, question that we asked was, can we have a hybrid regime where there's coexistence of non-steady state and steady state? And we found that, yes, we can, under special conditions where we basically have a symmetry going on with respect to B, which is like the middle kind of reactor, where basically the forces on it, if you think about it like that, are symmetrical. So it's remaining constant while A and C are kind of changing around it. So it's kind of like if you had an object and you were exerting a force in opposite directions that is equal, then you're going to have a constant velocity because it's a symmetrical effect on both sides. So just to kind of recap, the similarities are that time in physics and chemistry both have the same origin and the same kind of starting point of an event. And then clock time, again, can be used across domains. And then the new ones that we just kind of looked at is that there's transport time that's actually used in chemistry. That was kind of the special reaction which had a change of position involved. And, in both physics and chemistry, there is a mass conservation law. And then we also looked at the special invariances that occur within physics, within chemistry. So just to sort of wrap up, what I learned from doing this project is, I think, first of all, not so much like what I just talked about, but just how to communicate, again, with a professor. I think that will come, will be really helpful when I get to doing undergrad research in college. And then sort of how to learn, like we're given 6 weeks to really learn this focused topic and then also turn it into creative thought. So like kind of Robert was saying, it takes a lot of discipline, responsibility, and scheduling on your part to learn it and then think about it on your own, and then eventually come out with a 10 to 20 page paper on it. And then with that, obviously, how to write scientifically. And then, sort of at the bottom of the list is actually what I just talked about, just chemical kinetics. And so then I would just like to thank, would I recommend it? Yes, I definitely would recommend it if you're interested in like medical research or just med school in general. A lot of the guest speakers that come in twice a week, so I think there's 4 per week, are kind of oriented in that direction of medicine. And in my case, I'm not so much interested in medicine, but in like pure science. And I would still recommend it because, again, it's going to give you a leg up in that research experience. But I would say not so much if you're interested in engineering. I think there are a couple projects that are maybe computer science, and then I think there was one robotics project. But I think you won't get much out of the guest speakers. And but I would definitely look into it and see if there's a project that is interesting. So yeah, I just want to thank everyone, especially my professor. We basically called every other night for the entire 6 weeks and just talked about it, about the research. So that was essential for what I was doing. And then also as grad student, he helped me with a lot with like the graphs and the math, stuff like that. And then my writing mentor, and then my colleague from Ladue. So yeah, thanks.
They don't necessarily have a lot of say when they make a start. It's a very competitive program, and they might have thoughts and ideas of people they would like to work with, and they perhaps might not. So they kind of have to learn how to work with what it is, especially the year before, STARS didn't go. And this last year, it went under a limited model. Some of you were in person, like on God. Some of our guys were, and I think they did a great job kind of making the best out of the situation. And what I loved, Richard, you saying is, I don't know if I understood his PowerPoint. They dropped the information, and how many weeks before you kind of could go through that PowerPoint with some efficiency? 2 or 3 weeks. So half of his time was just trying to get a handle on what it is that he was trying to get done. All right, we've got one more presenter, and this is on our in-house programs. This is a trio. We've got 2 seniors and a sophomore. Guys, go ahead and take the stage. Matthew Light, senior JP Torak, and sophomore Thad Berta.
Hello, my name is JP Torak. I'm a senior here at SLUH, and I am joined by my classmate Matthew Light, who's also a senior. And we'll be presenting on some of the STEM activities that happen in-house at SLUH, the Applied Aviation Engineering class and the Robotics Club, which is partnered with the STEM Club. So this past summer, Matthew and I were students in the Applied Aviation Engineering course taught by Mr. Miller. The class covered the basic principles of flight from a physics perspective, engineering design, and then accumulated in building an RC plane. We also had the opportunity to visit several aviation-related sites, including the airport.
So the class actually began with kind of a discussion on the physics of flight and the 4 main forces, which are lift, drag, thrust, and weight. And we kind of talked about how those forces interact and what causes the forces, like the movement of air. And we kind of talked about how that applied to what our eventual plane would have to do and how to design that.
Another major section of the class was learning how to design and build the RC plane. So we started our designs by brainstorming ideas of what we wanted on our plane to include. And then we discussed, the next major thing we did was discuss the idea of biomimicry, or designing structures based on natural structures, particularly animal structures. So we tried to incorporate animal structures that either swam or flew into our plane, and that was really helpful for things like wings and then the tail control surfaces. We each picked an animal that flew or swam, and then we each designed our own plane on paper that had various aspects of that animal incorporated into it. And then we formed groups, and together we put all of our ideas together and came up with one design on the large paper that was the size of the foam board that the laser cutter was going to cut out the different shapes for us to build our plane with. After all of the designs were on paper were completed, we transferred them to Adobe Illustrator, which then went to the laser cutter, which cut out the shapes for the planes. After everything was cut out, we used hot glue and wooden barbecue skewers to build the shape. And then we put in various electronics, motors, and controls, and then programmed the electronics to the remote control that we use.
Also during this class, we got to kind of test out a few I guess, atypical technologies that aren't really usually present in an RC plane. I think the most major one is our leading edge slat design, where you could see down here in the middle. The basic premise of the leading edge slats is that so when you're coming in on a plane for like landing or when you're taking off, you have your plane pitched in the air like this, but the plane is moving forward like this. And what that creates is a difference between the angle the plane is moving and the actual angle of the wing, and that's called the angle of attack. And when the angle of attack is too high, there's not actually airflow over the top of the wing, and it doesn't. So there's no lift, and the plane stalls. So what the leading edge slat does is it takes air from under the wing and it redirects it over the wing, so you could have lift at a higher angle of attack than normal. So it kind of allows for more like slow speed and high angle of attack flight, and you can see that integrated up here. The black line's kind of the movement of the plane actually through the air, and the red line is more of the actual wing angle to that. And you can see how big that angle is between there, and it's still generating lift. Another technology are the Retivators right here, and it's kind of a combination of your elevators and like the big tail piece. So both of those control surfaces act to pitch the plane up and down and to yaw the plane left and right. And then the final one that's kind of big is this airfoil design. You can kind of see it here, called the Klein Fogelman airfoil. And what that does is it basically takes, normally from side on, the wing is kind of like curved. But with the Klein Fogelman airfoil, it has this big step down right here. And that basically uses like air turbulence to reduce drag on the wing. So we kind of tested it out. And the final product came out to be this. You can see our leading edge slats here, our Retivator, and the general wing shape right there. And we have a video to show you of it flying.
So this is a video from the last day of class when the plane flew.
Don't do some of your low flight nonsense right now. I'm not trying to, okay? Get away! Oh, all right. Hey. And then just bring it like. There you go. It's very Yep. You want to turn back this way? Okay. Oh, that just happened.
You can see from the video it likes to pitch up a lot, and that's actually because the center of lift is in front of the center of gravity, so it just creates a torque and it likes to go up, which is not supposed to happen, but you know.
So this class is offered to rising seniors every summer, and the way to sign up for it is during the, you'll be given a sheet or something when you're signing up for your senior classes in the spring. And so we're now moving to the second part of our STEM in-house, which is the Robotics Club. And so Thad is going to be joining us as well. So this is Robotics Club.
Hello, I'm Thad Berta. I'm a sophomore here at SLUH, and I do a lot of the programming for the robotics team. And so first is the organization that hosts the first robotics competition, which is what we compete in here at SLUH. This competition is a great starting point for anybody who wants to get into the STEM world, as building a robot takes just about every aspect of STEM. The parts of building a robot include, but aren't limited to, the design, manufacturing, assembling, and then of course the programming. First assists all the teams participating in the competition with standards for equipment and programming libraries to ensure ease in using that equipment. First can also provide mentors in the form of professional engineers or programmers to help teams build their robot.
At the beginning of January, we get our challenge for the first competition, and it usually takes 6 weeks from that until the actual competition. So during that time, we start off with having a big discussion, and we discuss, there's different tasks that earn you different points, and we discuss like what task we want to focus on to maximize our points. And after that, based on what we want to get points on, then we get our design. And so we start off with our digital design using CAD, computer animated design, and we do that on SolidWorks. So we kind of drop a 3D image or a 3D model of what our robot's going to look like. And then we get to the actual building. So that involves 3D printing. So we have 3D printers in the iLab and a robotics room, a ton of them. And 3D printers are very useful in getting prototyping and also different parts that will change a lot during the competition as you want to get like something just right. Then we actually do the actual building. A lot of it's out of aluminum, such as this arm here, and a lot of different motors and different gears and parts like that. Then we go into the electronics. So this involves programming as well as just laying out electronics of the motors, of the computer boards, of all the various systems that need to be connected together. And then we do our testing. So we need to be able to drive our robot. There is our test chassis to make sure it performs correctly, to make sure we have the actual skills to do what we need to do. And to help us with that, we have like a test bench to test various technologies and a test chassis like that.
So we have another video. This video is from with our latest robot in this past September and our most recent competition. And so the robot is climbing onto a pull-up bar that's on a pivot, so that when a robot climbs onto one side, it pulls the bar down, and then if another robot climbs on the other side, it'll even the bar. And the goal is to even the bar because that gives you more points in the competition than just one robot hanging. And so we kind of use principles from physics with like Mr. Baud of torque and rotational motion to balance the weights of the robots and to be able to climb up our robot to grab onto the bar, pull it down, and then when another robot came on, to be able to support our weight on that bar. So here's the video.
Second BG, we got 15 seconds remaining. Trying to see this remaining team.
So that was at the end of the competition. So that was a great end to the competition. And so my main takeaway from all of the STEM in-house activities is how important in the STEM field it is to work as a team. So even our robotics team, everyone has different interests and different talents. So Thad, like he mentioned, is really good with programming and electronics. Matthew is great with the CAD and designing, and my specialty is more on the building side. But when we all come together and use our different talents and communicate, we're able to produce functioning robots and functioning planes, and it's really cool.
So you may think it might be challenging to join the team, but actually any SLUH student can. So if any SLUH students want to, you can contact Mr. Hinders or Mrs. Wellen. So when I joined the team, I was able to learn from other team members that taught me. And if you're interested in any aspect of STEM, robotics has it all. So it's definitely a great place to nurture. So you can come learn with us.
The only thing the moderator said is don't run into anybody and don't launch anything. They've been very good. Guys, we're at the end of the evening. Mr. O'Keefe is going to close us out. I wanted to thank our presenters. I wanted to thank Mr. Anderson one more time. I wanted to thank our audience for giving us time this evening. I know everyone's got school tomorrow. Mr. O'Keefe.
I'll keep it brief. So the big thing I wanted to do is just thank everybody for coming. I want to thank Mary Russo, number one, for scheduling this not on the night of the National League wildcard game, because we scheduled it a long time ago, right? And then they won like 17 games in a row, so we didn't know it was going to happen. So it was perfect that it was tonight and not that night. But really, seriously, thanks. I want to thank Mary for all that she's done. This has really been her brainchild. We did this 2 years ago, back when the guys were doing stars in person, and we had different guys that were doing research down at St. Louis U. Then we had this thing called COVID that occurred. But early in the year, Mary came in and said, hey, I've got some guys in my AP chemistry that have done STARS this year. Maybe we should get this STEM night going again. And she's really done a great job at organizing all this, pulling it together. So I wanted to thank her. She's an amazing chemistry teacher. Done a great job as a curriculum coordinator as well this year. I want to thank Bill for all his work. Yeah. I want to thank Bill Anderson for all his work that he's done with his class. It's amazing the work that he's done in terms of teaching these guys in terms of environmental science, but also all the STEM and engineering practices that he's done in his class. And really just, like Mary said earlier, just our program at SLUH, I think is very strong for helping our students develop, having the fundamentals in biology, chemistry, physics during freshman through junior year, and then having all the different possibilities that they can take senior year really, I think, helps our guys develop very well. And then finally, the clubs. You can see from the presentations and stuff that the guys did here, a lot of the stuff that where they got the interest that started was in the different clubs, whether it be the STEM and Robotics Club, the Medical Careers Club. And then biggest thing I want to do is thank the students. You guys did an amazing job tonight. You guys were wonderful in terms of the detail that you were able to describe your different projects, and it's really inspiring. I look forward to seeing what you guys will be able to, do and the great things you're going to do in your life. I've been around here for a long time. I think this is my 33rd year. I've even lost track. But one of the coolest things for me is always seeing when the guys go out and the great things they do in life, and I can tell that you guys are all going to do amazing things. And then finally, just thanks to all the science teachers, math teachers in the crowd, the mentors at the projects, and then parents. I know how much important it is for you guys to just encourage your sons in terms of developing their science abilities, and I just thank you for that. So just overall, thanks everybody for coming.
I know folks have school tomorrow, but if anyone had any quick questions, feel free to stick around. Otherwise, thanks again for coming.